Axonal transport is vital for neuronal health and viability. Our protocol describes an elegant method for monitoring and analyzing axonal transport and can be adapted for various neuronal disease models. The use of microfluidic chambers enable precise spatiotemporal control, which is crucial for studying axonal biology.
Axonal transport abnormalities are implicated with several neurodegenerative diseases such as ALS. The microfluidic platform enables study of basic mechanisms as well as testing therapies to repair axonal transport defects.
The microfluidic chamber platform can be easily adapted to suit different aspects of axonal research, including axonal growth and degeneration of various neuronal subtypes.
This method is complex and usually taught hands-on. The visual demonstration will increase the accessibility of this procedure to any scientist interested in studying axonal transport.
Begin by casting PDMS in primary molds. Use pressurized air to remove any dirt from the wafer platform, making sure that the wafers look smooth and clear before proceeding. Next, fill a container with liquid nitrogen and prepare a 10 milliliter syringe with a 23 gauge needle. In a chemical fume hood, place the wafer-containing plate in a sealable container and fill the syringe with 2 milliliters of liquid nitrogen per each wafer-containing plate. Open a chlorotrimethylsilane bottle. Pierce the rubber cap with the syringe and inject the nitrogen into the bottle. Without pulling out the needle, turn the bottle upside down and draw back 2 milliliters of chlorotrimethylsilane